Hello, I'm Richard Young, your host with Facundo Batista and Lena Fayan for MIT's course on COVID-19, SARS-CoV-2, and the pandemic. We're about 11 months into the pandemic. In that time, a remarkable amount's been learned about the novel coronavirus and its immunology, epidemiology, and in clinical disease. This knowledge has led to accelerated development of vaccines, as you heard discussed in this course. And we anticipate the first lots of these vaccines may become available in only a few weeks. But there are over 500,000 new cases of COVID-19 being reported each day worldwide. This leads to today's subject, development of small molecule drugs, and a lecture by one of the world's leaders in drug development, Dr. James Bradner. Dr. Bradner is president of the Novartis Institutes for Biomedical Research and a member of the executive committee of Novartis. He served as an attending physician in stem cell transplantation at the Dana-Farber Cancer Institute and was a professor within the Department of Medicine at Harvard Medical School. Drug discovery and development's a remarkably complex and challenging process. The people who do this best are given a special name by their peers. They're called drug hunters. Jay Bradner is probably the best of these drug hunters. I know him as an exceptional leader, a scholar, educator, mentor, innovator, and a passionate advocate for patients. Jay, thank you so much for bringing your perspective on small molecule drugs to our course.
Wow, Rick, well, thank you so much for the invitation and your really overly generous introduction. I so enjoyed preparing for this opportunity to share some insights. It's fair to say in a space that's evolving very, very quickly, wonderfully for patients around the innovation of first direct acting small molecule coronavirus therapeutics. And as Rick has shared, I serve presently as the president of the Novartis Institutes for Biomedical Research. For all of you at MIT and Cambridge taking this course, just right next to Flower Bakery on Massachusetts Avenue in Kendall Square, which is just a serious discovery research environment. But it's fair to say, as the reports of the coronavirus pandemic were filtering back from our Shanghai site in January, we, like so many others, were caught unaware without an active program in coronavirus, needing to lean on technology experts in drug discovery, scientists in dengue fever working at our Novartis Institutes for Tropical Disease, to see what we could do to respond to this pandemic. But this is not a Novartis lecture. This is a reflection on the state of the art in small molecule therapeutics discovery. I have these disclosures to share in this academic context. And I suppose the most important disclosure is, as a discovery chemist, there will be molecules. And I am loath for any of these insights to be misconstrued as rules about drug discovery or therapeutics development, because there are always exceptions. So please take all this as a learning, but maybe not hard and fast rules. In fact, in the context of COVID-19, many of the rules have already been broken and most of them are being rewritten. First, just to ground us in the sobering reality, is to not lose sight of the ongoing profound burden of this pandemic. Here to orient us to the magnitude of disruption to lives, societies, healthcare, and economies is the latest from the Johns Hopkins dashboard with almost 60 million cases and 1.4 million deaths globally. In Massachusetts here, 200,000 cases, 40,000 active, nearly 200 in ICUs in this state. And I know many are joining from around the world, and we think about our associates at Novartis and societies in Belgium, Switzerland, Russia, the UK, and Italy that have so heavily hit in this second wave. And although many are frustrated and bored of this pandemic, we're not remotely out of the woods. And excited and optimistic and hopeful as I am about vaccines, I still see the need for definitive therapeutics for SARS-CoV-2. The impact of this pandemic has many faces, principally those infected, suffering from the illness, healthcare systems and healthcare workers on the front lines, risking exposure themselves and some falling ill, economic impact that's untold, as Triggs and Karras remind us, this unprecedented shock wave of demand shock, supply shock, financial shock with asymmetric impact on small businesses, low-income families, exaggerating an already pretty steep racial and social inequality, the most unequal recession in US history. And also there's great interruption to our lives in ways we don't understand: education, travel, and the social fabric. I seek to paint this picture because definitive small molecule therapeutics can remedy not just the virus, but restore society from the present pandemic and better prepare us for the future. On the flip side, what a time for science. And I know many of you are in the earliest moments of your training in science. Amidst all this disruption, I so admire the committed, inspiring open research that we've enjoyed in biomedicine. Self-assembling, collaborative, and transparent. Importantly, also frequently competitive, accelerating drug discoveries I'll share today. And I just cannot emphasize enough the heroic efforts of academic clinical investigators, global COVID research groups, 25,000 publications a day with a 20-day doubling time, more than 1,000 structures published to the PDB. Where many labs have rightly closed, like so many, our lights have been on with continuous R&D, working from home to keep our associates safe and focusing on a minimally essential portfolio, starting a new thread of discovery research for first small molecule inhibitors of the main protease of SARS-CoV-2, as I'll share in part today. We have overnight, as a community, created a COVID-19 therapeutic development arsenal, 777 unique compounds now in development, 311 in clinical study, 466 preclinical, and we have just 45 minutes together. So I will need to take choices around what to use to illustrate pathways of therapeutic discovery. And thankfully, while R&D is moving fast, viral evolution is proving rather slow. And this is effectively without currently the pressure of selection. This could change with vaccine and antibody therapies directed at spike. If D614G is a measure of fitness, what scares us is the possibility that there could be an acceleration in lethality, even to the level of MERS. And so we want to start then here today to skate to where the puck may go, to anticipate selective pressure of vaccines, to anticipate the next 8th zoonotic transmission of the coronavirus family. At challenging times like these, we really learn what an important vital industry we work in. All right, now I understand many in attendance here are not far along in their scientific training, and some surely are decidedly liberal arts concentrators. And I want to keep the talk engaging for both listeners, and that's a humbling assignment because there's so much sophistication in drug discovery and development. So let's start with basic. What is a medicine? It comes in a syringe, like a vaccine, a pill, like a medicine tablet, or biomolecules in a bag. In a vaccine syringe, this could be a large biomolecule, synthetic, aka lab-made, like an RNA, a protein mixed with immune-stimulating so-called adjuvants, or an inactivated virus, like the polio vaccine. In a hanging bag of saline on the right, maybe it's a biological molecule, an antibody, as in the lower right, a purified protein, or unoptimized small molecules that can't be tablets yet. For today, we're going to focus on the middle, what we consider small molecules. Maybe they're 500 Daltons or less. The majority of human therapeutics are small molecules. They could be natural chemicals like rapamycin, that ring on the left, or invented drug-like molecules like imatinib for leukemia next to it. These are as familiar as aspirin, Tylenol, statins, Prozac, and tamoxifen. They're collections of carbons, nitrogens, oxygens, and hydrogens, sometimes sulfurs and phosphorus, as I'll show you today, more frequently fluorines, rarely boron, someday tellurium. They're collections of atoms all stitched together in fashion, sculpted to occupy the binding site of a target protein of interest. And so let's get into the business of inventing a small molecule coronavirus inhibitor. You can think of drug discovery chemists like myself as molecular locksmiths. We try to shape a molecule, shown in orange on the right, to occupy fully a binding site, displacing water, driving energy so as to potently engage a protein, sometimes to inhibit it, sometimes to activate it. As shown here, it seems very static and rigid like a key, but proteins are actually quite dynamic, and aligning the ridges on the small organic molecule and its shape to be complementary to the atomic undulations of protein structure is highly technical, but it's fundamentally artisanal work. And the process is quite iterative. We imagine, prepare the next analog, test it to improve potency, binding specificity for the target of interest, but not similar looking or functioning proteins in the cell, biological selectivity to target only the pathway of interest or the organism of interest, and then eventually to engender drug-like properties. And that's medicinal chemistry. Shown here is a little blue molecule here, bound into its active site as well, in this case, a bromodomain. And you can see on the right all of the different parameters beyond binding potency and selectivity that we wrestle with when we're trying to take a lead molecule and make it a candidate medicine. We care about its solubility so that when it's taken orally, it dissolves. We care about its transport into the body and its distribution in all of the tissues. In coronavirus, it's particularly important that these medicines might make it to the lung, where so much of the disease pathology exists. We want the medicine to last a long time, and so we build it to be metabolically stable, and we want it to be safe. And so we avoid certain chemical groups on the molecule that are known to lead to end organ toxicity, but there's so much we don't know that we put molecules through a series of battery of tests in cells and in animals as well, to try to establish the safety that patients and clinicians expect from us. But we are sculptors, and here's shown the same molecule now, but with dotted lines that connect the partially negative charge on the molecule to the partially positive charge on the protein, or reciprocally the case. And we try to build in these contexts because every time we create a new so-called bond, we drive the potency of our molecule up. And as molecules become more potent, we can give patients less of the molecule, mitigating the risk of toxicity from the accumulation of a high dose of a drug. And that's drug discovery in a nutshell. Once we've discovered a medicine, though, we want to bring it into clinical investigation to test it. And I know this is the first mention in this course, but there are phases of drug discovery and development. The preclinical phase is where we establish the data set of toxicology and safety that the FDA and regulators around the world require before we're granted an IND, an investigational new drug application approval, to bring that medicine into patients for the first time, what we call first into human clinical study. And that's phase one, where we learn about the dose and the schedule. We learn whether we were right about how long the medicine lasts in the bloodstream and what it does to its target and its tissues. Once we believe we know the dose and schedule, we enter phase two. And this is where we expand out at 1 or 2 doses and we start to look for disease-specific activity. If the medicine continues to be well tolerated and even starts to show some signs of disease control, it might move into very expensive global phase 3 studies where we compare a single dose of the medicine to the established standard of care to try to look for drug efficacy, to see if this medicine really changes the practice of medicine. So how do we make these decisions? At Novartis alone, we have 291 drug discovery programs, 90 clinical stage molecules, 500 clinical trials, and these studies get increasingly very expensive. A phase 3 study might be $100 to $500 million. And so we prioritize this research at NIBR around these 8 dimensions of how big the unmet need is, which in SARS-CoV-2 couldn't be bigger. The tractability of making a drug. Is it an easy druggable protein or an undruggable protein? Will this molecule be first, or is there competition? And if there is, will it be different and differentiated from competition? Do we know or have a sense of all the possible patients that could benefit? What is the breadth of the opportunity? Is the target well validated, or do we need to do or collaborate to do genetic research to validate this target? And then what will this medicine do for patients in society? At Novartis, we focus on just transformative therapies. Medicines are going to have a monumental impact because it's the confluence of what society expects from modern medicine today and what the pragmatic business reality is, that there is today, rightly so, only a business for medicines that really move the needle. Now, this pandemic has been raging since the fall of 2019, but I want to calibrate that whereas vaccines research and certain biologics can be dramatically expedited because of mature therapeutic platforms, this artisanal process that I speak of in small molecules takes a bit longer. On average, small molecule drug discovery programs take 10 years, and in total, in aggregate, might cost $4 billion for every successful medicine, because the probability of success when starting drug discovery is about 5%. And I assure you, if there was a cheaper, faster way to do it, I'd be the first to sign up, especially my current role managing the portfolio of Novartis. So when we started to do drug discovery on coronavirus, we imagined that by resourcing this and cutting certain corners that you can cut, taking on risks that we normally wouldn't take, preclinical, never with patients, that we might accelerate that process. But we needed to think of our program as not just for SARS-CoV-2 and COVID-19, but for maybe COVID-25, God forbid, down the road. So how do we pick the target of our small molecule now that you've learned everything you need to know about building one? There's groups of proteins that are so-called druggable, meaning there's a strong track record of success at making a direct-acting therapeutic. These are proteins like enzymes, receptors, channels that have binding pockets. They have little pockets where cellular biochemistry is performed, where maybe they bind natural molecules, like the estrogen receptor binds estrogen. And so to then invent tamoxifen or to imagine a molecule that could bind that protein is easy to imagine. The G protein coupled receptors alone have over 130 proteins for which there are exemplified drugs. Unfortunately, SARS-CoV-2 doesn't have any GPCRs. The area of science and scholarship that I'm most interested in, and the program I lead scientifically at Novartis, focuses on expanding the reach of small molecule discovery science beyond what's druggable into the uncharted water of totally undruggable proteins or even RNAs for which there's never been a successful invented small molecule. I won't speak to this today, but new types of molecules that don't just bind, inhibit, bind, and activate as that key in that keyhole, but that can glue two proteins together to short circuit disease. Molecules that lock proteins in inhibited conformations, like our first SHP2 inhibitor, and maybe even molecules that target RNA. Because if you can target RNA in a sequence-specific way, well, then nothing is undruggable. And we indeed did realize the first sequence-specific engager of the SMN2 mRNA that stabilizes this RNA on what's called the SNRP, the spliceosome complex, stabilizing SMN2 expression to compensate when children lack SMN1. And we've recently learned that this same molecule destabilizes Huntington, perhaps a small molecule therapy for Huntington's disease. And it's not just us at Novartis. There's so many working on undruggable targets, and I make mention here of the remarkable work across the river at Vertex, who have realized small molecules that bind and modulate the CFTR channel for cystic fibrosis. Now, there's a great track record of antivirals that are small molecules, and so we have every expectation in approaching SARS-CoV-2 that we could be successful. Antiviral drug discovery is 4 decades in the making, more than 90 approved medicines, and many, many milestones. In the 1970s, ribavirin and acyclovir, the very beginning of antiviral chemotherapy, active, but not highly efficacious, not transformative. In the 1980s, this period was defined by the success of development of HIV antiretrovirals, establishing small molecule therapy as the definitive medicine combinations in HIV. First with AZT or zidovudine, and ultimately non-nucleoside reverse transcriptase inhibitors, protease inhibitors that would soon follow. And around 2005, we saw the advent of hepatitis C virus direct-acting antivirals, again used in combination, culminating in even curative combination therapy. The types of targets that people approach with viral drug discovery tend to be polymerases, large protein machines that create nucleic acids, RNA making RNA, or DNA making RNA, or DNA making DNA, and then proteases, enzymes that cut and cleave other proteins. And their success for both RNA and DNA polymerases, their success for both serine and HCV and aspartyl and HIV proteases, but many, many disappointments along the way. Many disappointments along the way. In particular, at approaching new types of targets like viral capsids or host receptor interactions. And though I won't speak much about biologics, they're few in number and only a few with a meaningful impact. And so we'll focus on small molecules here today. The biology of COVID-19 offers any number of great targets and points of intervention. I assume you remember the biology from Professor Glansinger's brilliant lecture a few weeks ago, and images from her seminal JBC paper in the spring are borrowed here to remind that this positively single-stranded RNA virus with its 30,000 nucleotide genome is loaded with therapeutic targets. It has a replicase complex that has a polymerase that's processive and proofreads. The virus has a very low mutation rate, as I shared, compared to other RNA viruses, suggesting that a molecule might work today and work tomorrow. That among the 13 genes encoded and the 27 proteins made, there are any number of good enzymes to target, including 2 proteases. And this main protease, or the 3CL chymotrypsin-like protease, will be the focus of one of our case studies here today. Each step of the viral life cycle might be considered for therapeutic vulnerability. And these viral proteins can be assessed for essentiality even before therapeutics exist, providing target discovery and validation. Now, there are many paths to finding a drug, like drug repurposing, taking a medicine for one disease and trying it in another. We call that molecular repositioning, to focus not on the vector, the virus itself, but on the host immune system for immunomodulation. And I've spoken already about vaccine development and neutralizing antibodies. We're going to talk principally about de novo drug discovery, but I want to illustrate, even with all the innovation in these other domains, why small molecule drug discovery is still very important. First, about repurposing. It's an experiment that should always be done, but it almost never works. To take the library of FDA-approved drugs and to test them in Petri dishes for their ability to inhibit a new virus and its ability to replicate and kill cultivated human cells. And it's fair to say some of the best scientists in the world in academia and in remarkable foundations like the Gates Foundation organized immediately to line up all the FDA-approved drugs and even clinical stage investigational drugs from pharmaceutical companies. We all contributed our small molecules, and unfortunately, the signals were quite weak. So though much celebrated, we have yet to really realize an active therapeutic through clinical drug repurposing. Still glad we did the experiment. You might know that until there are direct-acting and effective antiviral therapies, that what keeps patients alive are sedatives on ventilators, antibiotics to prevent ventilator-associated pneumonias, the medicines that nurses and clinicians need in the ICU to allow the body the time to repair itself. And these medicines came under short supply, even in the developed world, but especially in the developing world. And through companies like our own, with our generics manufacturing organization, Sandoz, we and others were able to assure supply to these life-sustaining medicines. But what we really need to do is to target the root cause of this disease. There are important signals that arose from the biological consideration, and I know Dr. Clark shared the clinical presentation of COVID-19 with many of these inferences. After infection takes hold in the epithelium of the lung tissue, it triggers an inflammatory cascade, elaborating many protein inflammatory factors called cytokines and chemokines, interleukin-6, IL-1 beta, IL-8, and TNF-alpha for the biology majors among us. This is a pleiotropic storm of inflammation. And the elevation of these factors, clinically revealed by ferritin, D-dimer, and C-reactive protein increases, become very challenging and very damaging to the patient. And so companies that had antibodies directed at some of these factors initiated clinical trials, as we did with interleukin-1 beta. But targeting a storm by taking out only one of the raindrops seems quite difficult. And so we and others have looked at targeting the nodal mediators of inflammation. And in the UK, a clinical trial was undertaken with a steroid called dexamethasone, not a getting muscular steroid, but a quieting inflammation steroid. And this intervention in hospitalized very ill patients was the first to demonstrate a survival benefit. by targeting a factor that switches on inflammatory genes and silencing it. These clinical investigators were able to keep patients alive longer in ICUs. Likewise, we are working to study other nodes of inflammation, like the NLRP3 inflammasome, and with our partner Insight, studying the intracellular signals of inflammation, the Janus kinases that are triggered after many cytokines. But despite massively parallel assessment of these abortive anti-inflammatories, we still have to target the root cause of disease. That brings us to vaccines. Dr. Corbett has already considered this in an extraordinary lecture I'm so glad I got to hear. But I think about Jason McClellan in the labs of Graham and Kwong, who reported through a decade of scholarship the way that one can stabilize this immunogenic spike protein that's used as the substrate protein for vaccines to raise a greater degree of immunity. And I think this innovation has defined the vaccination potential of mRNAs, such as the collaborative work with Moderna, adenovirus vaccines from J&J, Russia, and China, protein adjuvant combinations, where that very protein is given with immune stimulatory molecules, as traditional vaccines often are, that have yet to read out, but we're so hopeful, as well as inactivated viruses that bear this payload to hopefully trigger strong and durable immune reactions. As Rick shared in opening, there's incredible progress on vaccines, but we must be better prepared to treat the complications of COVID-19 and again, anticipate COVID-25. Now, Dr. Virgin in a week or so will be lecturing exclusively on blocking biologicals, so I just brought the one slide. This spike protein is not only good for raising immunogenicity, but also as a therapeutic target with proteins that might bind and put blinders on it so that the virus cannot find epithelial cells to infect. The spike protein engages with the ACE2 receptor on epithelial tissue, and one could imagine antibodies that would bind and neutralize this protein. Again, blinders on a coronavirus. I bring just one example here from a group in China who isolated antibodies from human patients convalescing, responding to COVID-19, and found a very potent antibody recently reported in Nature magazine that binds to the spike protein very potently and in a manner that prevents the receptor engagement. Shown in the middle of this screen in pink and purple is that antibody. Shown in cyan and yellow is the spike protein, and you can see the overlay of this gray ACE2 receptor. It can't reach the spike protein. And in animal models of this disease, rhesus macaques with infectious related lung damage, there was strong protection. These insights have taken groups like Eli Lilly and Regeneron and Vir forward to make blocking antibodies. There are challenges with the antibodies. They're intravenous or injectable. And so groups like ours and others have been looking for alternate proteins that one could use in earlier stages of disease. We have a collaboration with Molecular Partners, taking a protein fold, a mini protein called the DARPin, a designed anchor and repeat protein. Only 10 kilodaltons, these tiny little proteins can bind with incredible potency, again, to the spike, preventing the interaction of the virus with the healthy human tissue. But by having an array of 3 of these DARPins all stitched together, we get incredible avid binding like Velcro. And in animal models, here, the dwarf hamster model, there's a protection from death to coronavirus. It's very early days as these DARPins are just entering clinical trials, but as they're easily expressed in bacteria and can be subcutaneously administered, they're the type of medicine that won't have challenges with cold supply chain and could reach a lot of patients. So back to small molecules, there's a strong rationale to make an antiviral. It'll surely be complementary to any vaccine. Many people won't have access to or get a vaccine or will choose not to, which is scary. And as with influenza, even when vaccines exist, there's still a need for the sickest patients to have an antiviral. Plus, we must be prepared for a future pandemic. So in drug discovery, we actually don't start with the chemistry and the target. We start at the end with the patient, because the patient journey is really the North Star of our science when we're lucky enough to know who a medicine is intended for. This massively influences the design principles of a medicine. With repurposing or repositioning, you're stuck with the properties of the medicine you made for a different purpose. But most of the properties can be influenced. And so imagine on the left an asymptomatic child who might want an oral medicine. Maybe like my daughter's 9 years old might want a liquid medicine to drink. Imagine a symptomatic individual is in the middle who needs early intervention with combination therapy, not yet hospitalized. Again, an oral medicine could help. And on the right, patients in hypoxemic respiratory failure, it's fair to say might benefit from antivirals and anti-inflammatories. And so here for this project, we undertook to imagine treating all three of these patients. And that might take a molecule that's orally bioavailable and is active at all stages of the disease. Who is the patient? The second question is, what's the therapeutic hypothesis? As we start to understand disease with atomic resolution, not just the clinical symptoms and signs, but the fundamental organ physiology, the state of the cell, the mediators, the mediator proteins in the cell that trigger the disease. And we ultimately can visualize it with X-ray crystallography or cryo-electron microscopy as shown on the left to know where each atom of that large protein biomolecule is. We can see the keyhole and start to make a drug. And we build a hypothesis with infectious diseases. There's really a very clarified path because these are non-human gene products. And so we know that when we intend to inhibit the viral protein, we may not inhibit a human protein. There may not be one like it. So I'll talk a little bit about protease inhibition. And here's one such hypothesis. Irreversible, that means irreversibly bound, crazy glued molecule, substrate competitive binding to the SARS-CoV-2 cysteine protease might function to inhibit protein processing, leading to attenuated viral replication in the treatment and prevention of COVID-19. And these therapeutic design principles then govern where we go from here. Step 3, how will we discover the molecule? As Rick and I were chatting about as the course was starting, today's drug hunter is well equipped with established and also emerging and disruptive technology platforms. On the left, you see what we call the big yellow arm. This is a industrial strength robot that can do high throughput screening of 3 million compounds in a few days. Now we rarely need to go beyond 2000, but by one performing experiments in virally infected cells, we might find a needle in the haystack. For some targets, you need many more than even 3 million molecules. And if looking for a needle in a haystack, let's make a bigger haystack. And with today's what's called DNA encoded library chemistry, in the last 4 years, we've expanded the chemical equity of Novartis from 3 million molecules to more than 1 billion molecules. When we have protein structure bound by our molecule, when we can see the key in the keyhole, as in the middle panel, we can direct subtle changes and respond to needs. And starting points sometimes don't come from big molecules, they come from very small molecules called fragment-based drug discovery. And on the right, sometimes we're ignorant of what the target might even be. As in regenerative medicine, trying to grow cartilage or expand blood stem cells, we don't know how to do that. And so we use cell-based or so-called phenotypic screens to let the molecules show the way. Question 4, how are we going to develop this medicine? I told you the stages of drug development, but at each stage, there's an important learning that comes. I've listed these here. You can read them for yourself, but for COVID-19, we expect to address unmet need with transformative potential. And that might mean expecting or demanding from our science a benefit to overall survival as we get with dexamethasone. That this will improve on standard of care, that will give regulators hard outcomes around the world so that at the time when the medicine works, patients will know about it, doctors will demand it, and patients around the world may have access to it. This often involves thinking through things like supply chain, the cost of manufacturing, and working in a dialogue with payers to arrive at a fair and value-based price. None of these things are easy, but they're all part and parcel of drug discovery and development. And I put this last, but it's actually the first thing we do is we team up. The science of therapeutics is so technically nuanced. We're led to see it through the lens of molecules, atoms, measurements, gene names, lines of code. But the more I contribute to therapeutic discovery, the more I realize this is a team sport. This is a people business. I mean, if ever there's a team sport, it's drug hunting. As we say at MIT, this convergence of the biological and physical and actually medical sciences defines the experience of drug discovery. Also critical is the research context. There's a business structure function relationship that can't be underestimated. Is this a focus in thematic biotech with a clarified strategy, tranched investment, mercenary experts calibrated to binary outcomes in the roller coaster ride? Or is this a generational challenge that might better live in a pharmaceutical environment where revenues allow us this one advantage to work at a longer horizon of research? And in both settings, you need to partner to find expertise, critical technology, sometimes funding from BARDA, DARPA, Gates, and others, manufacturing scale, and then a path to bring your medicine to patients around the world. So I want to now share two real-world stories of COVID drug discovery. And this is the molecules part, so I hope that you're caffeinated. I'll talk through the successful innovation of medicines for the SARS-CoV-2 polymerase and a work in progress to invent first inhibitors of the SARS-CoV-2 main protease. So as Dr. Glausinger presented, the SARS-CoV-2 genome has what's called an RTC, a replication transcription complex. You'll remember all this from her lecture. It's a 9-protein complex that governs negative strand synthesis from the positive strand of RNA. And this is utterly essential to the viral life cycle to make more of its genome. It's an absolute requirement for vitality. And that makes it a good and compelling drug target. Now, one way to inhibit a polymerase that stitches little bits of nucleic acid together is to make a variant of a nucleic acid that would trick it, a molecule that would bind it more potently than the natural building blocks and thus shut it down. And this is called nucleoside-based drug discovery. And the discovery of remdesivir by Gilead is just a remarkable story of mechanistic repurposing. It was importantly a collaboration also with the US CDC, the Army Medical Research Institute of Infectious Diseases. Remdesivir is a member of the nucleoside class of antivirals. As shown in the lower left here, there's a base and there's a sugar. And it looks a lot like ATP, shown here with its triphosphates, and like GTP. And many successful antiviral drugs have been made by tricking polymerases with different looking nucleosides. The benefits is that these are often broad spectrum medicines. There's a high barrier to resistance, and it's just a stock pond of chemistry to learn from. But the risks are that these can be toxic molecules, in particular to human mitochondria, and they can have poor drug properties, often given IV. There was a long-standing collaboration between Gilead and the US Army to find drugs for Ebola. And the remdesivir chemical series, now published, was prepared in a nucleoside library expansion effort to target also the hepatitis C virus, NS5B polymerase. And you can see here in these little image, the path by which these molecules were made chemically, and this little R group. And this R group might be this cyano group, a simple methyl, or a more extended chain. By making these 4 variants, and then putting these molecules, 3A, B, C, and D, into a panel of viral assays, they discovered that molecule 3A, as I show here with my cursor, is a very potent inhibitor of SARS-CoV-2. And this greater than 30 micromolar suggests that it's non-toxic to cultivated human cells. And so this molecule fit for a culture dish was ultimately fashioned into a prodrug, a molecule that could be given IV, as shown here, and that is remdesivir. Note this triple bond is connecting carbon to nitrogen is this 1' substituted for all the chemists here, cyano group. And what this does is it tricks the polymerase, the viral polymerase. It gets inserted and installed into the replicating genome. But unfortunately, the chain terminates 3 nucleotides later, and the viral genome can't be duplicated. Now, though very effective, this prodrug has no oral bioavailability, and so it would need to be imagined as an intravenous medicine. The pharmacology of the drug's pretty cool. It's a prodrug. This little purple bit is shed as soon as the molecule enters the cell, and then ultimately it reverts to this free monophosphate version, and then phosphates are extended, and the triphosphate gets incorporated. A beautiful little bit of cellular chemistry. Well, this was invented in order to be an Ebola or hepatitis C drug, accidentally active against coronavirus, but chance favors the prepared mind. And so when the pandemic hit, scientists at Gilead collaborated with scientists around the world to put this molecule into the hands of people who had the viral isolates emerging from this growing pandemic. This is a crystal structure from a recent manuscript that shows the full ensemble of this RTC, this replication factory, bound to the template primer as shown in orange here, this strand of nucleic acid, and there in blue is remdesivir being inserted in as a chain termination. And that little cyano group that sticks its arm out over here serves to throw an elbow to normal human polymerases. And so it's non-toxic to human polymerases while very active for viral polymerases. The development of this medicine started a long time ago, but then moved pretty quickly in the pandemic. Research was initiated in 2009, as I shared. It was even tested in an Ebola phase 1 trial in West Africa in the DRC, but ultimately was abandoned as less active than some antibody therapies. But Gilead pivoted on a dime in order to make this medicine available in January of 2020 to scientists in China to test in isolates, in February of this year to expedite 2 phase 3 clinical trials. Jumping straight from phase 1 to phase 3 is a brave moment, but in the current pandemic, so appropriate. Advising the NIAID study called Active One and providing it to the WHO and INSERM as well. The clinical trial data so far is a bit of a mixed bag. In this first clinical trial reported back in April out of Hubei, China, a randomized controlled trial comparing this medicine to the placebo in a blinded way, 10 days of treatment, intravenous daily, failed to demonstrate clinical activity or a significant effect on recovery. But not to be dissuaded, a larger study was done to try to tease out statistical significance. And this active trial that surely Dr. Fauci presented in this course was completed in 1,062 patients randomized to remdesivir placebo with an improvement in median recovery time as shown up here on the, in this top panel, that the remdesivir patients recover just a little bit faster, 10 days versus 15. There's no survival benefit. There was no data on viral load. It did lead the FDA to provide an emergency use authorization and ultimately FDA approval. But since that time, a large World Health Organization solidarity study failed to confirm its benefit. So the clinical benefit is unclear. In some groups, like the WHO, are not recommending its use, but others, like the Infectious Disease Society of America, are for symptomatic individuals in the hospital. It's just a gorgeous masterclass in nucleoside drug discovery. And it's not alone. I bring this slide just to show in fair balance the efforts at Ridgeback Bio to innovate EID D2801, Molnupiravir from Merck now. This is an oral nucleoside analog, but here it's not an adenosine ring system. It's a hydroxycytidine ring system, so think C, not A. And it has a randomized control trial that should read out later this month. And another interesting molecule here from Atea, now partnered with Roche, AT527, is another oral nucleoside inhibitor, a double prodrug that actually makes a guanosine. And guanosine antivirals have been challenging from a tolerability standpoint. There's also unexpected preference for SARS over MERS, so perhaps not pan-coronavirus active. Well, in the remaining moments, I'll just introduce an early program at Novartis. This is to target the protease. The main protease is an essential gene that, as Dr. Glausinger shared, functions to cleave these beads off the string when the large polyprotein is translated from the viral mRNA. There are 2 proteases, one that's called the main protease, Mpro, another that's called the papain-like accessory cysteine protease, PLP2-Pro, but we'll focus on the main protease today. Compelling about this is that the protease is 96% identical in sequence between SARS-CoV-1 and SARS-CoV-2. So there is likely an evolutionary pressure for this target not to change. And here, as shown previously, is the function of this protease. As this long polypeptide is synthesized, the protease functions to liberate all of the viral building blocks. And it is, in fact, an essential gene. You know, the sequence of SARS-CoV-2 was only first presented in January, but already by March, 2 laboratories had published crystal structures of the main protease, showing us exactly what the protein does and exactly what it looks like. And what we found in the active site is a cysteine residue. This is a cysteine protease. This is, for the chemist, the little protein side chain nucleophile that cuts off one protein from the other. We got excited about this cysteine because over the last 4 years at Novartis, we had built a technology platform to drug cysteines. We've prepared libraries of over 1 million molecules that are trying to find cysteines anywhere in the proteome. And we search for these cysteines like people trying to, climb a mountain, Rick, as I know you do in your pastime. And here we're just trying to find footholds for drug discovery. And so working with scientists at Berkeley, Dan Nomura and colleagues, using very advanced technologies like mass spec and these very special, what are called electrophile libraries, we formed an energizing team. I joined the team myself in assay development to leverage this unique platform to find first protease inhibitors. We had to invent a lot of assays. Having pure protein was helpful. Knowing what it was looking like was helpful, but how would we measure in a petri dish the function of the protease in cells? How would we measure with purified protein the activity of the enzyme? And these assays, these experiments, are necessary in order to assess compounds and to improve their potency. We've now solved the crystal structure of the main protease from, it's fair to say, almost a dozen coronaviruses. And true to form, the overlay of one of these folds to the other is almost identical. I have a picture of it here showing SARS-1, SARS-2 in gray, and MERS in green. And look at the way that these strands are just superimposable from 3 separate experiments overlaid. They have the same keyhole. And this makes us believe that we could potentially realize a first pan-coronavirus therapy, an oral therapy that could be used for a short window of time in this and perhaps even future pandemics. Well, it's early days, but we've finished lead finding, and we're now into lead optimization, the artisanal process of improving the molecule. And though we don't normally talk about this science at this stage of development, we're also approaching this in a radically transparent and open way, inviting others to collaborate with us or even to compete with us. What could be better for humanity than a competition to cure coronavirus? And we should report here with these dose response curves already double-digit nanomolar, so really potent and cell-active proof of concept molecules from which we hope to make a drug. And this is the flow chart. We start with the chemistry. We then put it through biochemical assays. We use a lot of computer-assisted drug discovery to model and improve these medicines. In this battery of protein assays, we then pick starting points. The hit compounds then go around and around this circle where we change them, we test them in protein assays, we test them in cellular assays, and then we make new ones again. And out of this cycle eventually will come molecules that we start to believe could be drugs. And then we bring them into animal models of disease soon to come. This is called the target product profile. This is our North Star. This is how we intend our molecule to be realized. We hope it's oral. We hope it's potent and selective and well tolerated. We hope that this medicine could be used alone or in combination with remdesivir in sick patients with a monoclonal antibody, should they prove beneficial. And ultimately, because it would be oral and well tolerated, even early in disease as prevention or secondary prevention after exposure. We're not the only ones working on the protease. Colleagues at Pfizer, just a block away from us in Cambridge, took a molecule that they invented for human rhinovirus 14 protease and cross-tested it for coronavirus protease, just like Gilead did with their Ebola medicine, remdesivir. And though the molecule is intravenous and not very potent, by giving enough of this molecule, they're now conducting a phase 1 clinical trial in symptomatic patients to see if there could be benefit. There are a lot of great targets. There are other proteases like the papain protease. There is a helicase, an hexarabonuclease, a 2'-O methyltransferase, all of which Dr. Glaunsinger introduced to you. But whenever you see that word ACE in a protein target, that means it's an enzyme. And catalysis, enzyme functions, happen in these greasy little pockets where chemists like me love to position small molecules. So there's some fantastic additional targets that we won't have a chance to consider here today, not just in the virus, but in the host as well, such as the TMPRSS2 protease that spring loads and cleaves the spike protein to drive infectivity. One might make inhibitors of that protease as well. I don't mean to make it sound like everything's solved. For all of you that are students, we need you. The assays to support drug discovery are not suitable. They're not quantitatively rigorous. They're not highly reproducible, and that's slowing us down. And so better models in perhaps normal preclinical species like mice and better assay platforms are needed. We have to find a way not just to respond to the need in developed countries, but in the developing world, and find new paths to expedited development of these medicines, because as more somewhat active drugs are approved, really active drugs of the future will have to march through and compare them to advance them into earlier stages of disease. Suffice it to say, we really need and could benefit from your help. Well, I want to close on a high point here. The saving grace of this pandemic has been the massive technology investments that companies have made, truthfully for a different type of medicine, but that could be reconsidered at a moment's notice to imagine coronavirus medicines. In our case, covalent chemoproteomics. At Moderna, it's an mRNA gene therapy platform reconsidered as a vaccine platform, but this is not the type of preparedness that we'll need. In 2004, the Institutes of Medicine published a book that's hard to read today. It was on the heels of the SARS epidemic, only 774 deaths, and scientists organized to imagine what would be needed for when this coronavirus comes back. It is the playbook for what one might have been able to do had the thread of scientific innovation and drug discovery been maintained. And so I want to leave us all with this charge, that we commit to this science, that we find a way to make it sustainable. And even once, when we're finally out of the COVID-19 pandemic, we commit to delivering and anticipating the eventual return of this and other vectors. Rick, I want to thank you so much for the invitation again to participate. This course is quite a contribution amidst the upheaval in our personal and professional lives here, teaching middle school science as I do most evenings at our house. We've all appreciated this chance to learn aside you. And we've been watched 7.0 faster than the Queen's Gambit. And so thank you for that. And then lastly, to all dialing in who work in research capacity, you are unsung heroes of this evolving pandemic, managing personal challenges, overcoming the anxiety of the workplace, responding in a manner only you and only science can do. I so admire your work, your intentions, and your ideas. Thank you.
Jay, thank you so much for that presentation. Remarkably, as I and others were drawing out questions, you seemingly knew they were coming and answered many of them. But there are a few that I think students would be interested in that you haven't yet answered. How do you think about prioritizing the many targets among the targetable proteins in coronavirus?
You know, Rick, at the intersection of tractability, because this is an urgent situation, we don't want to take a generational strategy. We need to see immediate response to the pandemic. At the intersection of tractability and essentiality, the protease and the polymerase really rise to the top, And that's where we landed with the protease. We have any number of ideas how to target the proofreading apparatus that would lead to catastrophic incorporation of mutation, the methyltransferase that would have impact not just on genome stability, but perhaps also mutagenicity. But we, like so many others, responded with immediacy. When we're out of this pandemic and we start to think more liberally about scholarship in coronavirus, I do think some of these other targets will rise to the fore, even some of the presently so-called undruggable non-structural proteins of that genome.
You mentioned this extraordinary collaboration that's been happening. Do you view that as a short-term thing, or do you think that's going to have long-term effects on this industry?
Well, you know, I'm a real advocate and an active practitioner of an open and collaborative science. It's more fun to do science that way, and it moves much faster. And so there's so many reasons to do it. I think this pandemic is serving as a catalyst to change a lot of behaviors. Surely business travel, surely time with family. But on the flip side, in science, it has forced all of us to challenge our assumptions about open access publishing, about the use of preprint servers like bioRxiv, about sharing chemical equity. Would you believe scientists at Takeda, under the direction of Andy Plump, shipped us the crown jewels, the small molecules from their library, to a Novartis facility to test for protease inhibition? It's fair to say this sort of thing just never happened before. And we, when we assemble as the heads of R&D, as we do almost every single week through the pandemic, to brainstorm how to better care for our associates and respond to the disease, we wonder initially in jest and now quite seriously, what would happen if we approached Alzheimer's disease in this same, open and transparent yet still ambitious way? So I do think things will change meaningfully on the heels of the pandemic in the practice of science.
Jay, we're about out of time, but I just want to say, knowing that you left your academic work at Harvard and Dana-Farber to try to do something even more for patients, and it must just be an extraordinary ride being at this institution at this time. And we're grateful for all your efforts to keep us safe.
Well, Rick, I thank you for that. I missed time with you and in collaborating and with students especially, but this chapter is about therapeutics, and this, like many other environments, a wonderful place to work. And so thanks for letting me come back to campus for a day.
It's been a great pleasure.
Thank you.